Somewhere in southwestern Libya, there's a rock shelter you have almost certainly never heard of. Not because it is obscure, not because the science is complicated, but because the full picture only emerged in April 2025. And when it did, most of the world was looking somewhere else. In 2003, an archaeologist scraping back sand in the Tadrat Akas Mountains found something the Sahara should have destroyed thousands of years ago. Not fragments, not dust. Skin intact, leathery, unmistakably human. Two women wrapped in woven mats, dead for 7,000 years. At the time, nobody understood what they would eventually reveal. It would take 20 years, 3 separate research teams, and technology that did not yet exist. When the full picture finally emerged, it did not just rewrite the story of the Sahara. It erased an entire branch of humanity from the map, then put it back. For most of the 20th century, the scientific consensus about North Africa was straightforward enough that nobody felt the need to question it. The Sahara was not always a desert. Between roughly 14,500 and 5,000 years ago, it was something else entirely. 3.66 million square miles of grassland, dotted with permanent lakes, threaded with rivers, home to hippos and elephants and giraffes, and the people who hunted them. Scientists call this period the Green Sahara. The name is almost inadequate for what it actually was. The dominant model went like this. When the Sahara greened, it became a corridor. Populations from sub-Saharan Africa moved north. Populations from the Mediterranean coast moved south. Genes mixed. Cultures blended. The Green Sahara was a bridge connecting the two halves of the continent, the way a highway connects cities, and people moved across it in both directions for thousands of years. The evidence seemed overwhelming. In the Tadrat Akas Mountains alone, thousands of rock paintings showed cattle herders with bows, women grinding grain, children playing near water. Pottery found at Saharan sites matched styles from the Nile Valley and from the Sahel, hundreds of kilometers in either direction. Stone tools from multiple regional traditions turned up together in the same archaeological layers. You could draw arrows on a map showing populations flowing back and forth across a green continent. And the arrows looked exactly right. The model was published in textbooks. It was taught in universities. It became the default framework for understanding how North Africa was populated. And for decades, nobody seriously questioned it. There was one problem. Nobody had checked the DNA. This is the part of the story that deserves more attention than it usually gets, because the failure was not laziness. It was a cascade of reasonable assumptions, each one reinforcing the next. Ancient DNA work depends on preservation. Cold climates are ideal. Permafrost in Siberia, limestone caves in northern Europe, high-altitude glaciers in the Andes. Those are the places where genetic material survives for thousands of years. The Sahara is the opposite of all of that. Daytime temperatures regularly exceed 50 degrees C. Humidity is near zero. Ultraviolet radiation is relentless. DNA degrades fast in heat. And the Sahara is one of the hottest places on Earth. For decades, geneticists simply did not bother trying. They assumed, with good reason, that any genetic material in Saharan remains would be too fragmented to read. Then, there was the archaeological evidence itself. When you find pottery from the Nile Valley sitting in a Saharan campsite, the simplest explanation is that people carried it there. When you find cattle bones at a site where wild game was previously the only food source, the simplest explanation is that herders migrated in. Archaeology reads objects. It cannot read bloodlines. And for most of the 20th century, nobody had a reliable way to distinguish between an idea that traveled and a person who traveled. The result was a blank. North Africa became one of the least genetically sampled regions on Earth. Europe had hundreds of ancient genomes. The Near East was catching up. Between the Mediterranean coast and the Sahel, there was almost nothing. You cannot find a pattern in data that does not exist. And you cannot question a model when there is no competing evidence. The assumption was reasonable. It was also wrong. The reason Takarakori changed everything has less to do with who was buried there and more to do with where. Most Saharan sites are exposed. Open dunes, flat plains, surfaces that bake under direct sun for the better part of every day. Genetic material in those conditions does not degrade slowly, it disappears. Researchers had tried other sites across the region and found nothing usable. The heat was an effective and essentially total barrier to ancient DNA recovery, and the scientific consensus had adjusted accordingly. Do not bother looking. There is nothing to find. Takara is different. It sits inside a deep rock overhang in the Tadrat Akus mountain range, shielded from direct sunlight with interior temperatures that stay far below the extremes, hammering the exposed landscape outside. The two women were buried in dry sand beneath that overhang, and the combination created something that should not exist in the Sahara, a natural desiccation chamber, skin dried before bacteria could consume it, collagen in the bones held together. The same brutal environment that destroyed genetic material everywhere else had by geological accident preserved it. Here inside the densest tissues, teeth, and leg bones, tiny fragments of 7,000-year-old genetic material survived. Not much, not clean, but enough. In 2019, researchers managed to extract mitochondrial DNA from both women. Mitochondrial DNA passes only from mother to child, which makes it useful for tracing broad maternal lineages, but limited for the kind of detailed population analysis that answers hard questions. Still, even that narrow window hinted at something unexpected. The maternal lineages did not look like what anyone had predicted for this region. The results were strange enough to notice, not yet complete enough to act on. The real breakthrough came in April 2025. Natada Salem, a paleogeneticist at the Max Planck Institute for Evolutionary Anthropology in Leipzig, and senior author Johannes Krauss, the institute's director, published a study in Nature that changed the picture entirely. They had done what most geneticists considered effectively impossible. They had sequenced the full genomes of both Takara Korore women, not the mitochondrial fragment, the whole genome, the first complete ancient genomes ever recovered from one of the hottest environments on Earth. Salem described the process in interviews. The DNA came from teeth and leg bones, the densest tissues in the body, the ones most likely to shield genetic material from prolonged heat exposure. The fragments were short, degraded, and riddled with the chemical damage signatures that accumulate over millennia. Modern sequencing technology reads these fragments the way you might reconstruct a shredded document, scanning millions of tiny overlapping pieces and computationally reassembling them into something coherent. It is slow work. It fails more often than it succeeds. Entire research programs have ended without a usable result from far more promising material than this. But when it works, it gives you something that no amount of pottery analysis or rock art interpretation can provide. The actual genetic identity of a person who has been dead for 7,000 years. Salem's team had that. 2 of them recovered from a desert that was supposed to make recovery impossible. What those genomes contained was not what anyone expected. Salem's team compared the 2 genomes against 795 present-day individuals from Africa, the Near East, and Southern Europe, along with 117 ancient genomes from the same regions. It is the kind of comparative analysis that turns a single data point into an argument. You are not just looking at who these people were. You are looking at who they were not. The answer came back clean and immediately strange. The Takarakori women carried no sub-Saharan African ancestry. None. 0. In a region that the textbook model had placed at the center of a genetic bridge between North and South. These 2 women had no detectable connection to the populations below the Sahara. The corridor model had predicted mixing. The genomes showed none. Instead, they belonged to a lineage that had never been seen before. Not in any living person, not in any ancient genome on record anywhere in the world. The closest match was not found in any modern population. It was found in 15,000-year-old skeletal remains from Tafaralt Cave in Morocco, a site associated with the Ibero-Mauritian stone tool culture. These 2 populations were separated by 8,000 years and roughly 2,000 kilometers, and yet they carried the same deep North African genetic signature, equally distant from sub-Saharan Africans in every measurable direction. That is not coincidence. That is continuity across an almost incomprehensible span of time. The genomic analysis placed the divergence of this lineage from sub-Saharan African populations at roughly 50,000 years ago, right at the moment when modern humans were beginning the great dispersal out of Africa. One branch of humanity left the continent and eventually populated Europe and Asia. This group stayed, and they stayed isolated in North Africa for tens of thousands of years, while the rest of the human story unfolded around them. Geneticists call this a ghost population, a group whose existence had previously only ever appeared as faint statistical echoes in the DNA of living people. Indirect evidence of something that must have existed but had never been directly confirmed. Until two women wrapped in woven mats in a mountain rock shelter gave it a face. There is one additional detail worth sitting with. The Takarikori women carried Neanderthal DNA, but only around 0.15% of it, roughly 1/10 the amount found in modern non-African humans. Since Neanderthals lived in Eurasia and not in Africa, that trace could only have been acquired outside the continent. At some point very early, a small amount of outside genetic material filtered in through contact that left almost no other record. After that, the door closed. Then, there is the Green Sahara paradox, which is the part that genuinely surprised researchers. The lush conditions were real. The lakes and rivers and grasslands were real. Cultural exchange demonstrably happened. Pottery moved between regions. Livestock practices spread. And yet, the populations on either side of the Sahara did not genetically mix. Harold Ringbauer at Max Planck noted that this level of isolation is extraordinary, especially against the backdrop of Europe, where populations were mixing extensively during the same period. Culture traveled, genes did not. Ideas can move without people. The genetics say so directly. That is a result nobody was expecting to find. The Takarikori finding would be remarkable enough on its own. A ghost population, 50,000 years of isolation. A lineage with no living descendants. But it is not an isolated case. It is part of a pattern. And the pattern is showing up on every continent, where ancient DNA can reach far enough back. In 2016, geneticist Bastian Lamas at the University of Adelaide sequenced mitochondrial genomes from 92 pre-Columbian mummies and skeletons collected across South America, Bolivia, Chile, Peru, Argentina, and Mexico. Remains ranging from 500 to 8,600 years old, selected specifically to represent the broadest possible geographic and temporal range. His team identified 84 distinct maternal lineages, 84 separate lines representing the full genetic diversity of the ancient Americas. Not one of those lineages exists in any living person today. Let that settle for a moment. 92 individuals, 84 unique lines, all extinct. The researchers did not need complex statistical modeling to understand what they were looking at. They compared the ancient sequences against modern genetic databases and found nothing. No matches, no descendants, no survivors. The diversity that had existed across the ancient Americas before European contact was not reduced. It was erased. The ancestral history behind those lineages adds another layer. The populations they descended from had split from Siberian groups around 23,000 years ago, spent roughly 6,000 years isolated on the Bingian land bridge between Asia and Alaska, then expanded rapidly southward around 16,000 years ago. After that initial expansion, communities settled into geographic pockets and stayed. Maternal lineages became distinct and localized across generations. After European contact in the late 1400s, most of those localized lineages simply vanished. Lamas was direct about what the data showed. Some of those populations were wiped out completely. Then in 2025, a separate study published in Science Advances reported the first ancient genomes ever recovered from Colombia. A team led by Kim Louise Charetta at the Senberg Center for Human Evolution analyzed 21 individuals from the Bogota Altiplano, some dating back 6,000 years. 7 of the oldest belonged to a population that had never been documented anywhere. Their DNA matched no known ancient group in the Americas. It matched no modern population anywhere on Earth. By 2,000 years ago, they had vanished entirely, replaced by a genetically distinct group with ties to Central America. Charit was precise about what happened. The genes were not passed on. There was a complete exchange of the population. One detail worth noting here because it connects to something broader. The oldest artificially mummified bodies on Earth are not Egyptian. They are the Chinuro mummy of northern Chile. Dating back 7,400 years, a full 2,000 years before the first Egyptian mummification. Coastal fishers who embalmed everyone, including infants and unborn children. Their specific mitochondrial lineage almost certainly does not survive in any living person today. 3 continents, different timescales, different causes, the same result. Entire populations alive for millennia, coherent and culturally distinct, gone without leaving a single genetic trace in the people who came after them. The survivors were not the whole story. They were a selection, and we built everything we thought we knew around them. The evidence deserves honest handling. And honest handling means saying clearly that not everyone finds it as conclusive as the headline suggested. The sample size objection is legitimate. 2 individuals from one rock shelter cannot definitively represent the population history of an entire region. North Africa is vast. The Takarakori women came from a single site, a single community, a single moment in time. Extrapolating from 2 genomes to a continental population model requires assumptions that the data itself cannot fully support. Researchers who have spent careers mapping human genetic diversity across Africa have noted this carefully, and they are right to do so. There's also a geographic explanation for the isolation that does not require dramatic narrative. The Green Sahara was not a uniform landscape. It contained mountain ranges, permanent lakes, dense wetlands, and ecological zones that may have functioned as natural barriers between communities, even during the wet period. Populations could have remained genetically separate simply because the terrain kept them apart, not because of any deliberate or culturally enforced boundary. Isolation does not always need an explanation more complicated than geography. For the South American data, the picture has a similar caveat. Sampling of modern indigenous populations across the Americas remains incomplete. It is possible, though with every passing year increasingly unlikely, that some of those 84 lineages survive in communities that have not yet been genetically tested. Absence of evidence is not always evidence of absence, and the databases against which ancient sequences are compared are still growing. Savino Darnia, the archaeologist who first excavated Takarakori, noted something that complicates any simple narrative of total isolation. The community buried in that rock shelter was not culturally sealed off from the world around them. They traded. They adopted hearing practices that originated elsewhere. They participated in regional exchange networks that connected them to communities across a wide geographic area. Culturally, they were engaged. The question the genetics forces is more specific and more unsettling than whether they knew their neighbors. It is whether the people who shared their ideas ever shared their bloodlines. In this case, the evidence says no. That is genuinely surprising, and it deserves more investigation before anyone treats it as settled. What is not in dispute is the 35,000-year silence. The lineage diverged from southern African populations around 50,000 years ago. They appear clearly at Taperalt around 15,000 years ago. Between those 2 points, there is almost nothing. An entire chapter of a people, unwritten, possibly unwritable. Where did they shelter during the last glacial maximum when the Sahara expanded to its greatest historical extent, and the conditions that sustained human life would have contracted sharply? How many communities were there? How far did they range? Johannes Krauss put it plainly. We do not know where they were between 50,000 years ago and 15,000 years ago. What ended them is less mysterious. The rain stopped, the lake dried. The savanna turned to sand, a lineage that had survived ice ages and climate shifts. And the entire Neolithic revolution went silent as the green world that had sustained it collapsed around it. Not through conquest, not through absorption, through climate, slow and absolute and indifferent. Nadasam's team is already searching for more ancient genomes across the Sahara. The odds of finding preservation like Takarakori again are slim. The desert does not give up its dead easily. The instinct when confronted with findings like this is to reach for a framework that makes them feel manageable, to slot the ghost population into a revised diagram, update the model, redraw the arrows on the map, and move forward with a slightly more complicated version of the story we already had. That instinct is wrong. What the mummies are revealing is not a correction to the existing framework. It is a challenge to the framework itself. The human family tree is not a clean branching diagram. It never was. It is a tangle of lines that split, drifted apart, briefly reconnected, and in many cases disappeared entirely without leaving anything behind. For most of the history of this science, we built our models around the branches that survived, because those were the only ones visible. The survivors shaped the story. The lost population simply did not appear in the data, so they did not appear in the thinking either. What ancient DNA is forcing slowly, and with considerable resistance from established models, is a reckoning with how incomplete that picture always was. The branches we lost may have been just as numerous as the ones that endured, just as widespread, just as old. The Takaracori lineage persisted for 50,000 years. The unnamed Colombian population had been on the Bogotá Alto Plano for millennia before anyone replaced them. The 84 maternal lines of the ancient Americas represented a diversity that took tens of thousands of years to develop and was gone within generations of European contact. These were not marginal populations clinging to the edges of the human story. They were central chapters in it. We just could not read them until now. The technology that made Takaracori possible is being applied to remains that would previously have been written off entirely. Sites in hot climates, in humid environments, in conditions that conventional wisdom said could not preserve readable genetic material. Some of those attempts will fail. Many already have, but the threshold of what is recoverable has shifted, and every shift expands the territory where ghost populations might still be waiting. Every blank space on the ancient DNA map is now a potential ghost. North Africa had almost no ancient genomes before 2025. Colombia had none. There are entire continents and time periods where the data simply does not yet exist. And the pattern that is emerging wherever data does exist suggests strongly that the blank spaces are not empty because nothing was there. They are empty because nobody had found a way to look. The corridor model for the Sahara may not be entirely dead. Cultural exchange happened. That evidence is real, and it is not going away. But the genetic picture is now far more complicated than arrows on a map ever suggested. And the confidence with which that model was held for decades looks in retrospect like a function of ignorance rather than knowledge. We did not know what we did not know. Now we are beginning to find out. That process is not finished. It is barely started. The ancient DNA revolution is not a conclusion. It is a method. And what it is methodically revealing continent by continent, site by site, is that the human past was larger, stranger, and more populated with vanished lives than anyone had previously had the tools to imagine. Return to the two women in the rock shelter. They were not symbols. They were people, herders, weavers, members of a community that had persisted longer than most civilizations can conceive of. They did not know they were the last chapter of a 50,000-year story. They buried their dead carefully in sheltered ground, wrapped against a world that was already drying around them. The science that found them is not finished. Salem and her team are still searching. The 35,000-year gap is still silent. There are almost certainly more ghost populations waiting in rock shelters, in mountain caves, in the compressed sediment of dried lake beds across the Sahara and beyond. We do not know how many branches of the human family tree ended without descendants. We are only beginning to learn how to ask the question. The mummies are the answer. We just have not yet learned to read all of what they are saying.